Hello, everyone. My name's Philip Brooks. I'm from Telluride High School, and this summer I was lucky enough to spend 6 weeks at the George Lab at UC San Diego, and that's the lab that studies the neuroscience of addiction. So, in our lab, the main experiment is the gene-wide association studies, and essentially what it is a study that tests addiction predicted genes in rat models, and they are hooked up, as you can see, to a saline solution containing either cocaine or oxycodone, and they are given a lever that they can press if they wish, and we're studying their behavior and their neurological pathways, and through this experiment, we could theoretically predict which rats are going to be addicted and decide from there if it can translate to the human population. So while there wasn't a gene-wide association study going on while I was in the lab, I was lucky enough to assist my mentor with her PhD experiment, which was a dose response of nicotine in mice. And we're essentially just looking at how different concentrations of nicotine affect the mice in their behavior and their neurological pathways. And so, so they're put in these administration chambers, and essentially a vapor that we create ourselves is pushed into the chamber, and it'll either be a 0, 5, 15, or 50 milligram concentration of nicotine. And after about an hour, they are moved to an open field test, which is essentially just a box with some walls. And it is there to assess their anxiety levels. And basically, the more they stay on the edges near the walls, the more anxious they are because they don't feel as safe. Following the open field, there is the more gruesome part of our study, including the perfusion, where we have a fixing agent that's pumped through their system, and it basically just flash freezes their brain, keeps it as a snapshot of what the nicotine's doing in there so that we can study it later. And then we remove them, we extract their blood, and take the serum out of it to determine the levels of nicotine that were actually in their system. Following that was my favorite part, in which we got to mount and image the slides. And so you first get to slice up their brain into very thin slices, about 30 micrometers, which is thinner than the width of a single hair. And after that, you are meant to mount it onto a slide, which takes a lot more delicate touch than I had, and I ended up ripping at least 30. I think that was the first one I ever got. I was so proud. I whipped my phone out right there and then. Following that, we would stain them with this chemical that attaches to the cells that have been activated by nicotine, and it stains them brown, as you can see in that top left corner. And after that, we're able to image the brain, and as you can see, there's all these little lights, and each one of those is a cell activated by nicotine. And once we have that, we can kind of see which areas of the brain are activated and what that means as to which parts of our body and our command center it is affecting. Outside of my work, I enjoyed the wonderful array of options I had in San Diego. Definitely one of my highlights was a skydiving expedition I embarked on, where I did. No, there was no monkey business. I kept my legs firmly inside the plane. I got to see some gorgeous sunsets right outside my house on the cliffs. I was lucky enough to spend time with some of the other Pinterns in the area, and we got to go to the San Diego Zoo. I discovered some unbelievable new food, and if you can believe it, that's actually an acai bowl. But I only ate that once because I felt pretty unnatural and toxic, you could say. We also had the pleasure of watching a SpaceX rocket launch right over our house. It was right at sunset, so everywhere was dark, but the rocket was so high up in the sky that its trail was still illuminated by the sun. I got to do some, I don't know if I'm allowed to say it. I did some cliff jumping. Very dangerous. Sarah's not supposed to know about it, but I got to watch some more sunsets. Got to put my cooking skills to use thanks to Trang, and I made my very own truffle fries. And I got to end my internship with a wonderful performance by a local performer, Andy Citron, right on the beach. And although I never had any pictures, I made sure to surf or body surf every single day of my internship. So my advice to future Pinterns, at least for me, I decided to hop on the website of my lab, and I read about 6 words before I realized I didn't have any idea what I was going to be studying. And I just want to tell the future Pinterns to not stress. Obviously, know a bit about what you're going into, but if you don't know everything, don't worry, they're not expecting you to. I think everyone should minimize the time you spend in your room and always try to go out and do something. You're in a new place, and you'll never know what's really out there until you go out there. Don't feel too frustrated if you're not immediately going into things. I couldn't relate because I was just giving rats drugs right from the get-go, but I understand that some internships slowly incorporate you. And lastly, if you have host parents, make sure to express your gratitude with actions as well as words. Sure, people like to hear thank you, but doing your dishes, giving them gifts, things like that, making them dinner, that's what they really appreciate. I don't have time, but I would like to say thank you to all these people and to you.